EuroScipy.
Okay, hi everybody. So firstly, thank you very much for giving me the opportunity to speak here today. So I'm going to talk about pyFRET, which is a library that I have made for analysis of data from single molecule confocal spectroscopy. So firstly, just to give you a really brief introduction to this experimental technique. So what is this single molecule confocal spectroscopy? Well, it relies on a physical phenomenon called forced resonance energy transfer. What is this energy transfer? Well, if you choose to have two fluorophores that you have chosen so that the donor spectrum, the excitation spectrum of one fluorophore overlaps with the emission spectrum of the other fluorophore, then you can effectively excite the donor fluorophore and see photons emitted from the acceptor fluorophore. And this is a distance-dependent effect. So if you look at the graph on the top left, you can see that when the two fluorophores are very close together, you get very efficient energy transfer. So almost all of your excitation is emitted in acceptor photons. But when the molecules are far apart, when your fluorophores are far apart, you get very little energy transfer, so very low FRET efficiency, we say. And so this effectively gives us a molecular ruler that allows us to measure distances within individual molecules by labeling them and looking at the ratio of donor and acceptor fluorophores that we see. And you can use that for studying things in biology like protein folding and protein aggregation. So that's the physical phenomenon. What does the experimental setup look like? Well, we have something like this. So we have a really dilute solution of molecules that we have dual labeled. And we fire a laser through that really dilute solution to basically excite the donor fluorophore. And then we have two photon counting devices that collect photons that are emitted from the donor and from the acceptor fluorophores. And then we can look at the ratio of those to see, how far apart are these fluorophores. So when I said dilute, I meant really dilute. We're talking about kind of picomolar quantities of, or concentrations of our molecules. So if we look at this over here, and this shape like this is our Gaussian laser excitation beam, we can see that we have some molecules diffusing around in solution, and when they diffuse through the confocal volume, then you see emission, and you want to choose your concentration to be low enough that you have hopefully at most one molecule in the confocal volume at any one time. And so in fact, most of your experiment is waiting around for something to diffuse through your confocal volume. And here's some data to demonstrate that. So we have the donor the donor photons at the top in blue and the acceptor in red. And you can see that you're kind of waiting and waiting and waiting, and then bang, you get a donor fluorescent event, and then you wait a bit longer, and then you get bang, donor and an acceptor fluorescent event. So now you have a data analysis problem. You want to take this raw data, and you want to select out the events that correspond to molecules diffusing through the confocal volume. You want to get rid of things that are just noise, and then having found these fluorescent events, you want to get rid of any residual noise in those time bins, and then you want to process that data to go from raw photon counts to some molecular distance information. And so that's a data analysis problem. What did that look like when I started my PhD? Well, it was a bit of a mess. So all of the different research groups that use this technique use their own hardware, they build their own microscopes, they have their own file formats, they have a wide range of different tools for doing data analysis, and they do things like give you code on a USB drive and say, you can try it, it probably won't work, which is great. So I made pyFRET, which is an open source library for doing data analysis in this field. It supports doing confocal measurements, and it supports a couple of different types of data collection methods. So this continuous excitation, which is where you just have your blue laser, your donor laser on all the time. And you can also have a mode where you have two lasers that flash on and off very fast, which give you some information about whether your molecules are properly labeled or not. So what does it do? It does all of the things that I was describing before. So starting from your raw data, you can do event selection and denoising. You can calculate the efficiency of transfer. You can export data as CSV files for further processing if you want to make, really fancy graphics. And you can also make some simple graphics using pyFRET itself. And in the interest of keeping simple things simple, like Ben Nuttall was talking about this morning, this is essentially the complete script that you would need to do that kind of data analysis. So you can see that first you have a list of files that you read in. You initialize a FRET data object, and then you can do file parsing, you can do background subtraction, event selection, removing crosstalk, which is another form of noise, some data manipulation, and then you can make some plots like these FRET histograms that I was talking about. So what do these plots look like? So this one at the top is what's called a FRET efficiency histogram. So along the x-axis, you have the efficiency of energy transfer between your donor and acceptor, and on the y-axis, you have the number of events that showed that efficiency. You can also make some scatter plots and some two-dimensional plots. You can make heat maps, and you can make 3D plots of either the raw data or some of the processed data later on. And does it work? Yes, it does. So we took a series of molecules that were DNA molecules labeled with a donor and acceptor, where we knew the distance between the dyes, and we had basically an increasing set where you increase the distance between the molecules, between the fluorophores. We analyzed some data from each of these DNA molecules using pyFRET. We fitted them, and then we managed to recreate this FRET efficiency curve that we saw on the first slide. So yeah, it does work. And then just to finish up, pyFRET is an open source library. It has unit tests. It can be downloaded from the Python package index PyPy. Here it is. It has documentation on read the docs. And the simple stuff that it does well. It could do more stuff. It could always do more sophisticated event selection algorithms. It could have, a way of taking configuration files, but for what it does, it works. So that's it, short talk. Thank you very much for listening. I'd also like to thank my PhD supervisors and my PhD funding body, who give me lots of money to waste my time making open source modules, and to everybody in my research group who's let me borrow their data for testing this, and in particular, actually, to Magnus, who was the first user and told me all about how it didn't actually work. Thanks.
Thank you very much for a talk that must be inspiring for every person doing research, I think. Any question? We have time for 1 or 2 questions.
Thanks for the interesting talk. Some time ago, I had a conversation with a Cambridge University-based chemist who runs some open science meetups. He suggested that in chemistry there is a particularly bad ecosystem of open research and sharing data and methodologies. Does your work here mean that that's beginning to change?
I don't know. I mean, my suspicion is that, I'm writing a paper about this at the moment, and I'd like to, publish it, and I'd like to encourage other people to, contribute code to this, because I think it would work well, or at least to use it as a benchmark against which to test their techniques. But I think what will happen is that I will release it, and somebody who is, a big shot professor in this field in the US, I'm going to be quite honest here, is going to say, this is useless because it doesn't implement my algorithm, which is the best one. And you're like, well, that's great. Maybe you'd like to contribute your algorithm here. And it's quite interesting that this kind of lack of openness can also be used as a way to shut down other people's research, to reject a paper or to insist that, it goes through another round of review because it didn't use a particular technique that somebody else used. But if you don't have access to, what they do, that doesn't necessarily just mean, re-implementing their code. It might mean building a new microscope, calibrating everything, and then doing this kind of data analysis and, software development for that. So there is a problem, and it's not just a software problem.
And a related question, is there a journal in your field that is open to papers about software and that welcomes such contributions or not?
So I think PLOS computational biology certainly would. That would be an appropriate field for this because it's kind of on the overlap between kind of chemistry, biology, biophysics. So that would work well. It is computational biology. For more specifically chemistry stuff, I don't really know. I think you'd have to kind of position yourself really in the physics field in order to get traction.
Thank you very much.